So good morning all. So we welcome you on the lecture number 24 of our GAN course. So ma'am will now Professor Kavita ma'am will start the lecture on the topic immunotherapy, precision medicine in action, CAR-NK therapy and dendritic cell therapy. So we will start our lecture.
Thank you, Leknath. So, although the topic says that we will start with CAR-NK and CAR-dendritic cell therapy, but in reality, I will tell you more about CAR-T therapy, many different kinds of CAR-T therapy, and that is really a lot of fun. Okay, so just to remind you about CAR-T therapy, so in CAR-T therapy, you draw the T cells from a patient, you draw the blood actually, then you separate the T cells and then you engineer the T cells. So you add the DNA, and we talked about how you can add the DNA to the cells. There are many different ways, and then the DNA makes the protein, and this protein is actually CAR, chimeric antigen receptor. And then once it is expressing, you where is number 3 here, 1 2, then you make tons and tons of CAR T cells, you expand them, you infuse it back to the patient, and then these are ready to recognize the antigen and kill the tumor cells. Okay, so for CAR T things, which T cells do you use? So usually you use a 1:1 ratio of CD4 CAR T cells and CD8 CAR T cells. Okay, so you use 1:1 ratio. These are also called as helper CAR T cells, and once, see, these are the chimeric receptors. So they latch into these tumor cells, and then they will release these perforin, cytokines, granzyme. Granzymes are serine proteases. So perforins create pores, and then granzymes get inside. It will chew up all the protein. The tumor cells will die, and once they die, they release all the antigens. So other immune cells are recruited in that area, which will clean up and attack the tumor cells furthermore, cleaning up the tumor. Okay, so just to give you a little bit idea about CD4 and CD8 cells, so these are both T cells. So they have found, scientists have found that 1:1 ratio of CD4 versus CD8 is the best combination. So CD4 plus CAR T cells provide cytokine support and has CD8 functions, and they are helper and cytotoxic. They secrete these ILs, and CD8 are also called as cytotoxic tumor cells, T cells. So they are involved in direct tumor killing. They release perforin and granzyme, which leads to rapid tumor death. So usually 1:1 is the best ratio for enhanced efficacy and improved persistence. You want these guys to be there for a long time. So the CAR T therapy is good in a way that these T cells are in human body. Once they are infused, although it is very expensive, we talked about it yesterday, but once it is there for a very long time. So now I'm going to tell you a lot of fun stuff. So this technology, once it was made, then people are making all sorts of combination. Okay, so these are special variants, and these are not all the variants, just a few variants. There are a lot more than these, but this will give you an idea. So if you take a simple CAR, so simple CAR is this, and this is, as you know, this is an antibody. So this part, so in the antibody, this is called the variable part. This is the part which recognizes the antigen. Okay, so this part and this part. So this part is equivalent to this part in CAR. This is the one which will recognize the antigen, and then all of these things are there, because if it just binds and does nothing, it is useless. So these domains are added so that it recognizes, and then it transmits the signal to kill the tumor cells. That's why you have these domains here. Okay, so first kind is tandem CAR. In short, it is called TAN CAR. Okay, so look at this card. So this is so tandem means it recognizes two antigens simultaneously. So if it recognizes two antigens simultaneously, the binding will be tighter and it will be more specific. So look at the CAR here. So it has two parts to recognize. So usually it has only one part here, right? But look at this thing, it has one part and another part, and these two parts are recognizing two different antigens. Okay, so the important thing is that how do you differentiate between TAN CAR and, so this one is not a TAN CAR because this is a CAR T cell, but it is expressing two different receptors, altogether two different receptors. So you can imagine one of this here, one of this here. This is not tandem, this is bisystonic. So I am one CAR T, but I am expressing two different receptors, one for this antigen, one for this antigen. That is bisystonic. But if there is only one CAR, but it is recognizing two antigen, that would be tandem. That's the difference. So that's what is shown here. So look here, this is one CAR, but it is attached to two different recognition domain. So this is tandem CAR 1 and 2, part of one only. This is bisystonic. Although CAR T is one, the cell is one, but it is expressing two independent receptors. Okay. So this is tandem CAR T. So what happens is that anytime you may even make a CAR T, right? So tumor will try to hide that neoantigen, okay? Because it knows if the antigen is expressed, it will be killed. So if you have two recognition sites, the second one will still latch on, right? So that's the advantage. So tandem CAR recognizes two antigens simultaneously. See, first part, second part. This is TAN CAR. You can even make three antigen specific. This will recognize one antigen, second antigen, third antigen. So triple. Now this is not TAN CAR because the same cell is expressing two different ones, the whole thing. This is called dual CAR. This is called triple CAR. But if there is one receptor altogether but different domains which are recognizing, that will make it tandem, yes. Yeah, so here for simplicity's sake they are showing that there is hardly any linker, but usually they provide different linkers, and when you are developing this thing, what you do, you try many different combination, you change linker length to see. So this is a very important thing whenever you make tandem. So suppose this is the tumor, right, and you are making two antigens specific, but one antigen is here, one antigen is here, and if your linker is not flexible enough here, it is not going to bind here, right? If it is next to it and if the linker length is flexible enough, then it will bind here. So that is always the case, that how do you know what is the exact linker length? But you have to give it some flexibility so that it can see the whole surrounding area. Having said that, what people do, that some people say, oh, we are making a TAN CAR where it will get activated only when it binds both the antigens. But then the problem is that it binds one antigen, but the second antigen is not in close proximity. So this is rendered ineffective, right? So it is better. You will obviously get more specificity if it is able to recognize two tumor-specific antigens, right? But that is also comes with a risk because they may not be next to each other, or your linker may not reach it. A linker is way too flexible to just find something next to it, right? So usually it is better that it works even with one recognition. Makes sense? But you have to play with anytime you make tandem kind of stuff, you have to play with the length of the linker to identify the best linker. Okay, so two linked, these domains are called SCFV. So two linked SCFV domains, like this is again CD19, CD20, then rest of the things, they remain the same. So advantage is that only one CAR construct, it is relatively cheaper to make and easy to manufacture, less risk of tonic signaling than two separate CARs, strong synergy when both antigens are present. Okay. Now dual CAR, as I said, dual CAR is this one. This is not. Here you are putting two different ones at the same time. That is co-administration. You are giving two drugs at the same time. So this is not dual CAR. You are just administering two independent ones simultaneously. Here also you are mixing them together. See, all of them are separate. But here this is bisystonic or dual CAR. The single one is expressing two independent receptors. Here the advantage is that they are likely to bind. But here you have to also play with the concentration of the dual CAR, right? If you use too much of it, then every CAR will just bind one of the antigen because you have huge excess of it. So you have to play with the concentration of this one so that both of them are used at the same time, right? Keep in mind, one CAR will actually not have only 2 copies. They will have multiple copies. So you have a choice of getting both of them lashed on if they are at the right distance. Okay. Then this is very good, modular CAR T. This is very, very good way of doing it. And you will see why. So what happens is that tumors are very adaptable, right? They keep changing their tumor antigen. So you spend $500K in dollars, right? $500,000 just to make a CAR T therapy against an antigen, and that antigen, the tumor hides that antigen after some time. So the whole money just basically goes as a waste. So this is a really awesome way of addressing that. So modular CAR T is an advanced form of immunotherapy. So you look at this thing. So this is the tumor cell, and these are the antigens, the black ones. Okay. Forget about this part for a minute. Okay. This is the CAR T here. Okay. So this CAR T will recognize this green circle here. Okay. So you can make the green circle separate. This is not directly attacking the tumor cells. It is only binding this thing. And then based on the antigen, you can change this structure. Okay. So imagine, let's give the analogy of the Gyan course, right? You are all the students and I am the teacher who is teaching it, right? But I did not know any of you and I had no access to any of you. And no. So Venkatesh is the middle person who knew me, who got me, and he's the person who recruited all of you, right? And if he organizes another grand course, he will recruit other students, and same teacher or a different teacher. So he's the one who is coordinating between you guys and me, right? So this part here is Venkatesh. You guys are whichever one you want to choose. One of us is here, and all of you are. You understand the point here, right? One part, second part, and the middle person, right? So the beauty is that Venkatesh can choose whoever he wants to choose as a teacher. He can choose whoever he wants to choose as the students, right? So this is very versatile method. So what happens is that this part is fixed. You make a CAR T. And you don't have to go for which tumor antigen, nothing. Everything is ready off the shelf. And now tumor cells will try to be very smart. They do different kinds of things. So you just change this. This part is ready because this part will bind here. This part is ready. You just change this part according to the tumor antigen and it will work every single time. Is it making sense? So this is shown here. So in between you have CAR T and these are all tumor cells. And then, so you give this modular compound. Okay, so this is the CAR T here, okay, and then you give the switch molecule. So on one end it will bind this one. So let's look at the green one here. So green one is binding the CAR T receptor, and the top part is recognizing the tumor antigen. Okay.
These switch molecule are generally small molecule or also?
They are small molecule, relatively small. Let me see, tumor antigen. No, it will not be a small molecule because it has to recognize the antigen. So it has to have a whole receptor-like a structure to identify the antigen. So it would not be a small molecule because they are not receptors. If they are receptors, then it can be a small molecule. So for example, if it is HER2 receptor, definitely small molecule there.
Yeah.
So it can be a small molecule. Most likely it is a small molecule because these tumor antigens are not displayed. So what happens is that its tumor cells are intact cells. So they are not, they most likely you are targeting the receptors there. So you can always use the small molecule like folate there, right? So it is definitely a small molecule. So this is the beauty here, right? So you are recognizing, and CAR-T of course is a receptor. So small molecule link together. Okay, so if you use low concentration, you kill one cell, then more and more you kill all of this, right? Makes sense. Now this is even better. Okay, so look at here. Look at these tumor cells. I'm showing two of them. Even in this one, they have different receptors, the blue receptors, the black receptors. And you can also think the tumor is heterogeneous. So someone will have blue receptors only, someone will have black, somebody will have yellow receptors. All different kinds of receptors, right? So you add this compound. So this green part is engineered to recognize the CAR-T, and this pink part will recognize this blue receptor. So this one dies, okay? Now here, this one is blue receptor, this cannot recognize. So this one will not die, but then you switch it to a different one. See, you switch it to different one. So now this one will die. If you use both, now both populations will die. So the beauty is that your CAR-T is the same. Okay, CAR-T is the most expensive part to make. You are just switching molecules. As tumor switches, you switch the molecule. And linking two small molecules is a piece of cake, correct? Okay, yeah.
We will have to do repeated biopsy to know if the antigen is.
The chances are CAR-T therapy is so good that when you start it across for some antigen or some receptor, it will kill it. But suppose it develops resistance, or if the tumor is heterogeneous and you do the biopsy, you already have 2-3 things in hand, or maybe 5-3 things in hand. So you can easily make this switch molecule, right? Because these are two small molecules linked together. So you can easily make it, okay? Now keep in mind that suppose this CAR receptor does not bind a small molecule, it binds something, but we know what it binds. You can even express that protein and link it together with a small molecule. That's a piece of cake too. So if the tumor is heterogeneous and you have already 5 candidates, you can make 5 switch molecules. And then CAR-T are there for a long time. So if you're targeting multiple things at once, the chances are the tumor will be eliminated. But if it comes back, you can again do the biopsy a year later or something.
And the switch molecules we are providing, they are provided systematically, like orally or intravenously, or they are.
CAR-T therapy is given intravenously because all the biologics, they are not given orally. So depending on what kind of switch molecule you are giving. So if you're
cells, they are basically cells.
So it could be 2 small molecules linked together. It could be a protein linked to a small molecule. So depending upon the stability, you can decide. If it is, if it is just a simple small molecule, maybe you can give it orally also. And it will be non-toxic because where will it go and bind? It is tumor specific. And one part is specific to CAR-T. Yeah. Go ahead.
Ma'am, that switch is occurring inside the cell, or we are giving 2 different compounds which are having different chemistry together?
No, chemistry, it has nothing to do with the chemistry. So let's say that I am the switch molecule, right? So I am there. So there is one party here, one party here, okay? So suppose I hold Shivani and Janvi like this, and I switch, then I hold Janvi and Venkatesh, okay? And I do Venkatesh and Vanshika. So I am changing, I am the switch molecule and I am changing what I am holding. So I am the same, but my hands are different, holding different people here. So this is changing. I mean, humans can hold anyone, so this is not the best analogy, but basically you can bring any two partners together. One is CAR-T partner, which I'm keeping constant because I have to get the CAR-T, so that ligand is always the same. So on one hand, I always have CAR-T. Here I can, if I change this, I can bind different partners. Okay, and what are these different partners? These are different tumor antigens. So I have the receptor for the tumor antigen, or I have the ligand for the tumor receptor. Make sense?
So when you have to bind to the receptor of that cell, you will like express ligand. T cell will express ligand. When it has to bind to the receptor,
No. It's not like that. So CAR T cells are ready to go. Okay, they have the receptor, they have everything. So CAR T is just there. You don't change anything in CAR T. So CAR T is standing here, ready to go. And you know they have the receptor, this SCFP receptor. So I have to have the ligand to bring CAR T here. So this is the ligand which is holding onto CAR T. I'm the switch molecule, correct? I'm not changing CAR T, I'm just holding on to that, okay, by non-covalent ligand interaction. So I have the ligand, CAR T doesn't have the ligand, and then I have the ligand to bring the tumor cell, or if tumor cell has a receptor, but if it is expressing some ligand, then I have the receptor there. Is it making sense? Yes, absolutely.
in this case, we are using just two small molecules linked together, and we are using T cells which are endogenous. They are not giving
We are using T cells expressing CAR T receptor, chimeric antigen receptor. Yeah. So you are right. CAR T is not standing on its own. It is part of the T cell. Because it has to use T cell machinery to spray, pepper spray to tumor cells. Yeah, you're right. That part is clear. So now you guys can see how awesome is that switch molecule. Tumor cells can keep changing their thing, and you can keep changing the switch molecule. Okay.
Some CAR T cells have their life inside the body, so we are binding them to switch molecules outside the body, or we have once injected the CAR T cells and then we are switching the switch molecules.
It can be given along with CAR T cells, and it can be given right after the infusion of CAR T cells. The best thing is that to give them together, but once you give CAR T cells in the body, they are there for a long time. So you can also keep injecting these molecules to the patients. I don't know what is the half-life of these switch molecules, so maybe they need to be injected periodically to the patients.
Yes, they will be cleared.
They will be cleared, right? So you have to periodically inject that, but that is a piece of cake. CAR T is there. CAR T expressing T cells are there. Let me say it like that, right?
Ma'am, the affinity of the CAR T cells towards the switch molecules, when we find out that the antigen the first switch molecule we provided, the antigen for that is now not expressing, and tumor is expressing other antigen.
Yes.
So we inject a new switch molecule.
Yeah.
So we will have to make sure that its affinity towards the T cell is more than the previous switch molecule. So T cell will.
So if let's say this is T cell, and this is the T cell. This is the chimeric CAR, right? Chimeric antigen receptor, right? So this is going to remain the same, right? So the small molecule which will bind, or whatever part which will bind, will remain the same. We just have to change the top part which is binding the tumor cells in case it changes antigens.
But that one will be kept, it will be binded to the CAR T receptor. So when we
No, so like Professor Venkatesh said, the small molecules will be cleared off by the body. So the older ones for which the tumor antigens have changed their thing, they will not stick around for that long, for years, right? T cells are there for years, so they have to be given periodically So the old ones will be cleared and you can give the new ones. Okay, but let me ask you, suppose they are expressing something which is not a receptor, how will you make something which will bind there? Anyone, can you tell me? So suppose the tumor is expressing some kind of weird hill-like structure, like some antigen on the surface. How will you recognize the tumor with that? If it is a receptor, it's okay. Maybe you can make a molecule. But how will you recognize some kind of protrusion unique to the tumor cell?
Is it in the cell or just a callus, like the whole tumor?
It is a part of a cell. Tumor is just, just the tumor shape will not do anything because that will change obviously. Like it is something sticking out of the cell.
Then how do we recognize it?
How will you make something in the switch molecule so that it recognizes it?
That day you said that antibodies kind of molecules can.
You can make a nanobody. Wow, guys, good. You can make a nanobody which will recognize the tumor. You have to get CAR-T next to it. See, now you have all the things in your arsenal. Just use, take out this, take out this. You already know that. You don't have to switch, keep a small molecule there. You can attach a nanobody there, right, to recognize it. All right, now we come to voltage logic gated CAR-Ts. Okay, add, or not circuit. So what it means is that when it is an add circuit, so this CAR-T will recognize the tumor only when it binds two antigens, A and B. Okay, if it binds A, it will not work. If it binds B, it will not work. If it recognizes both A and B, then it will work. Okay, so that will give high specificity. But again, the linker part is very important here, right? Or either A or B. Okay, now let's look at this. Not is sometimes the best one. So this is not A. So suppose there are two antigens, A and B. Okay, and I want you guys to think about it. So if it binds A, it will not work. If it binds B, it will work. If it binds both A and B, it will not work because A is not. So it can only bind B. So why it is extremely useful? So let me repeat it. There are two antigens, A and B. Okay, so CAR-T will only work if it binds B. If it binds A, it will not work. And if it binds both A and B, then also it will not work because A is not. Okay, so why is that important? How can you use it? Why people have even made such kind of CAR-T?
It's specific to the natural cells.
That's right. A is very good. A is also expressed in normal cells. So that is why if it is binding A also, then it will reject it. B is specific for tumor cells. Very good. That's why the NOT gates are very important. Okay. So this is here. OR gate. OR means either A or B. Okay. So then it is more versatile. If tumor is heterogeneous, our thing is also heterogeneous. At least it can recognize two different antigens. So it binds A, it works. It binds B, it works. If it binds both A, B, it definitely works. And means alone A is not enough, alone B is not enough. Both together it works. C is just the NOT gate. So when it binds A, it doesn't work. When it binds when it binds A, it works. When it binds B, it does not work. When it is bound to both A and B, it still does not work. Okay, so and this is because one of them is expressed in healthy and that is the NOT signal. Okay, so I'm telling you, so this is very important for tumor heterogeneity because not one, if you are just counting on one, it may not be expressed or tumors may hide it. So that is why it is important to have the AND or NOT signal. Okay, and if you are checking two things together, they are likely to be more specific. So if it is a NOT signal, you are sparing the healthy cells. Okay, and it also works in immunosuppressive tumor microenvironment. Okay, so let's look at the AND gate again. It will only work if both A and B are working because it's AND. So it doesn't depend on A and B alone. It has to have work when A and B are together. So this is the OR gate, either A, either B, both still works. If is, so let's go to NOT and AND. AND is, this will not work with bound to A alone, not work when bound to B alone, AB, together it works. NOT gate, here B is the problem. So if it is bound to B, it will not work. If it is bound to B alone, it will not work. A and B are bound together, but since B is involved and B is also expressed in normal cells, it will not work. But only A, which is specific to tumor cells, it will work. So basically what they are trying to tell you that normal cells also express A to some extent, but they will also express B, but tumor cells will not express B. So you are making it extremely specific for tumor cells. So these are logic gated ones. If better, so here when it binds to A, it works both ways. When B is there in tumor, so tumor cells A, so that is great. If better gate, that is not commonly used, but here A is there, B is there, it is still working a little bit because A and B, because you still have A, but here B is completely absent, so this is normal, so it should not work here, because, so problem is that A is expressed in all three of them. Okay, B is expressed in tumor cells. So this is a mixture of everything. So this is not a very good option because you will also kill the normal cells in there. So you have to make clear NOT signal. Okay. All right. OR gate means either A or B. Okay. So you already know this thing. NOT gate you already know. So NOT gates are also called as ICARs or inhibitory CARs. Okay. So ICAR detects antigen on healthy tissue, for example B. So if they recruit, if they see if they see antigens on healthy, they said no, we are not going to attack it. So they bring inhibitory signal. When they detect tumor antigen, then it's a yes signal. Okay, so that is what it is. So it prevents toxicity to vital organs expressing low levels of tumor antigens. Okay, so this is a very good example. Antigen A, PSMA is expressed in tumor cells. Healthy tissue express EpCAM. So, but healthy tissues can also express PSMA. So they will kill only when PSMA exists without EpCAM. So healthy tissues are completely spared, only tumor tissues die. So this is an example. This is inhibitory CAR, so it will not recognize the healthy antigen. This will say, okay, no, kill. So if both of them are active, then no one will win the game. If it says no, then no. Okay. When this is switched off, then only this will kill it. That's ICAR. Okay. So this is that inhibitory CAR. Okay, so now you can see why logic gating matters, off tumor toxicity and not improved specificity. When it is OR, then the advantage is that you have made against 2 tumor antigens. So at least it will bind one of them, so it will keep working. Okay, tumors are heterogeneous, so more A, B, C, D, E, F, G you put it there, better it is, right? But it has to be all tumor specific. So even if tumors are hiding the antigen, something will bind. Okay. So, and ICARs are very important to spare the healthy cells. Now let's look at armored CAR T cells. These are cytokine secreting T cells. So armored CAR T cells, what they do, so this is a CAR T cells. So once it recognizes the tumor, it actually releases a lot of cytokines and recruit other immune cells. So it is like, it is just saying come, 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 and the moment it says tumor cells, it will try to secrete cytokines, and different kinds of cytokines recruit different kinds of immune cells. Someone will recruit macrophages, someone will recruit NK cells. So basically all these cells will come close to it and kill the tumor cells. So if the tumor cell environment is immunosuppressive, it will overcome that. Okay, because it is releasing the cytokines. So these are different kinds of cytokines which are released. IL-2, IL-12, when it is L stands for?
Interleukin.
Interleukin. So IL-12, when it is secreted by CAR T cells, activates NK cells, macrophages, Th1 cells. IL-15 enhances CAR T persistence and memory. Okay, that is very good. You don't want CAR T cells after spending $500,000. You don't want CAR T cells to disappear from the blood. You want them to be there. So IL-15 keeps them going. IL-18 converts cold tumors to hot. How would you define cold tumor versus hot tumor?
Tumors are susceptible to receive treatment. They are
Cold tumors are susceptible to receive
resistant.
They are resistant. They are resistant to receive tumors. Why? Why?
They are less aggressive,
They are-
less attacking.
They are less
Attacking. They are more what you call inactive kind. Like
more than
they
that.
are not as rapidly dividing
So
as they
you are saying they are non-aggressive. That is not necessary.
comparatively less.
They are most in a quiescent state of action.
It seems like cold just gives us an idea that they are quiescent, quiet, non-aggressive, but cold tumors are those tumors where immunosuppressive cells are absent. So, oh sorry, not immunosuppressive, immune cells are absent. So, all these T cell, B cells, NK cells, they are not there. So, they are in like real nice tumor suppressive environment, just enjoying their whatever it is. Okay, so if you have more immune cells infiltration, those tumors are hot tumors. Okay, and they are more, if you target them, they're more likely to be targeted because you have immune cells floating around. Cold tumors have very few immune cells floating around, not immunosuppressive, immune cells floating around. They are resistant to therapy, like she said, because there are no immune cells there. So cold cells are not necessarily non-aggressive ones. They could be aggressive ones, but they're also deeply buried in their own immunosuppressive environment. So nothing can reach them. Is it making sense? So they appear cold outside, but they can be pretty bad. But the thing is that if you think it in another way, if it is aggressive, it is going in all directions, then other things will also come to that. So in that respect, they won't be cold. But so that is why the definitions varies a little bit, but one way of looking at it is less infiltration of immune cells.
So, ma'am, presence of only immune suppressive cell in their microenvironment makes them hot cells, hot tumor cells.
Immune suppressive. That is what I was saying wrong. When I meant immune cells, I was saying immune suppression cells. So say it again. What exactly do you mean? Immune cells or immunosuppressive cells? Cold are those which have immunosuppressive environment and they don't have infiltration of immune cells like T cells or NK cells. Okay? All right. So this is what they are doing. These armored CAR T cells, they release cytokines and get the help from other immune cells. Okay, CAR NK cells are very important and they are becoming very popular. So we are always comparing CAR T cells with CAR NK cells. So CAR is the same. If you put it in NK cells, it is NK cell CAR therapy. If you put them in T cells, it's CAR T. Okay. So CAR NK cells are engineered natural killer cells that express CAR similar to CAR T cells. But there are some differences there. So remember, NK belongs to innate immune system. And what is an innate immune system? From birth, and which is instantaneous. There is an attack, instantaneous. Okay, so that is innate. And naturally killed tumor or virus infected cells without prior sensitization is just immediate thing. Okay, so and we know that they originate from lymphoid cells. What are the cells? What are the immune cells which originate from lymphoid lineage? This is the kind of question you're going to get it in the exam. B cells, T cells, and NK cells. So lymphoid lineage is B cell, T cell, NK cells, correct? All right, so these are from lymphoid lineage. So look at this thing. So this is very interesting, the way NK cells recognize self versus non-self. So it is just really interesting. So remember I told you that every nucleated cell in our body has MSC1 and they keep showing their peptides to that. So NK cells actually recognize the MSC molecules. By the way, do you guys know what is HLA? Some of you are immunologists. So can you tell me what is HLA? Did you register yourself as immunologist? Otherwise I'll direct it. You are immunologist, right? Okay, okay. So Shivani is going to answer. That's okay, don't worry about it. If I'm put on spot, I'm blank too. Okay, so MSC1 in humans, MSC molecules, they're 1 and 2, right? 2 classes. In humans, we don't call them MSC. Our MSCs are called HLA, they are identical. These are human leukocyte antigen, nothing but MSC. If you call MSC, okay, human MSC1, human MSC2. But humans have also given their own HLA name to it. So you cannot use HLA for a donkey or some other organism. They are MSC. All vertebrates are MSC, but we are the same MSC in us. It is called HLA. Okay, so here is the thing. Forget about the antigens. So NK cells recognize our HLA or MSCs. Okay, so they will. So my MSC is different from your MSC and your MSC and your MSC. So we all have unique MSCs. So your NK cells will recognize your MSC and my NK cells will recognize my MSC. So MSCs, so all naturals, every nucleated cell in the body have MSCs there. So NK cells are surveying and they recognize our MSC, and that's a switch off signal. That tells us it is self. Right? But cancer cells, they are trying to be very smart, so they downregulate the MSC. So the MSC negative signal does not go to NK cells, they will kill it. Do you see that? Suppose, yeah, that is not a good analogy what I was going to say, but you get the point here. So if you downregulate MSC, NK cells will kill you. You are supposed to show up. Okay, so suppose you are coming through IIT gate, you better show up your picture. If you try to run, then they are going to follow you and put you somewhere else, right? So go through the proper surveillance process. So this is that signal here. So what happens is that this MSC, so this is NK cell, this is a healthy cell, and these things are MSC molecules. If it was human, we will call it HLA. So they are expressing, so NK cell says, oh, this is MSC. They say, oh, this is self signal. They will spare it. Now you make them disappear. These guys have made it disappear. So they said, oh, no MSC, foreign thing, kill it. So it is killed. That's how NK cells are surveying. Okay, so this is normal cell. They are showing MSC. NK cells find it and they said, okay, great, this is there. Okay, but if MSC has some foreign thing, then it will recognize that MSC is same, but the foreign particle is there, then they will kill it because then they know there is some problem there. Tumor infected cell, they are trying to be smart. They take MSC inside, now it will kill it. Okay, so that is the way they survey it. Okay, this is yet another one. So if they recognize that the MSCs are missing, they will spray it and they will kill it. Okay, so now if you make a CAR T cells near tumor cells, it's a very bad news for the tumor cell, right? It will just really kill it up. So you isolate T cells or NK cells. If you want to make NK CAR, you isolate NK, otherwise you isolate T. You insert the chimeric receptor gene, you expand them, you infuse it back, and now it is in the bloodstream. Okay, and now they will take care of the rest. Okay, now I'm sure you guys are very curious about what are the differences between CAR T and CAR NK cells. So there are some pros and cons on either side. Okay, so these are innate immune cells, you know, they spray perforin, granzyme and all of these things and kill it. And this is minimal GVHD risk. So this is something I have written here. GVHD is graft versus host disease. So suppose ideally it is very expensive to take the T cells or NK cells from the patient itself, right? and then engineer it and give it back to that. So suppose you take it from a healthy donor, right? So what are the things which are recognized by dendritic cells and all of these things, this HLA receptors. So if you remove all the HLA receptors, then it will not be recognized. But if you don't change it, then what happens when you give the T cell therapy or NK cell therapy from a healthy person to a patient? So this is a difference. So the cells which are coming will attack. So the cells which are coming from outside, the CAR T cell, they will think these are the cells inside are foreign. These guys will think you are foreign. They will kill each other. Cancer will survive. They will just kill each other. Okay. So when outside cells attack the patient thing, that is called GVHD. So for NK cells, GVHD risk is minimal because you are using NK cells which do not have a strong immune response against that one. But for CAR T, if it is allogenic, that means it is coming from a different patient, not from the same patient, then it is much higher. We have fixed that problem, but this is inherent problem. Okay, so allogenic works for NK cells, but for this you have to isolate it from the patient and put it back in the patient. But the good news is that this stay for very long time persistent. This has very short life. This can go for years, this can go for 3-4 weeks to a month or something like that. Okay, so weeks, and this is sorry, did I write the time here? I have not written, but NK cells don't stay for very long. CAR T cells stays for very long. This is CRS, ICANS. CRS is cytokine release syndrome and immune effector cell associated neurotoxicity. So if you give CAR T cells, body can rebel against it. Okay, and body can say, oh, foreign thing is coming. So these things are those things here. Let's not go in the detail. But for CAR T therapy, this kind of side effect is severe. NK cells, it is very low. GVHD is very rare for NK. If you give it from the patient to the same patient, no problem. If you take a healthy donor, it's a problem. See, ideally you would like to take it from healthy donor and just keep a lot in stock. And anyone needs it, you give it to the person. But then there is this problem. Allogenic use means someone else's thing. It's very limited. This is excellent. So all of these things, they have pros and cons. Okay. So limitations of CAR-NK, although safe and potentially cheaper, CAR NK cells face challenges, poor persistence. They don't stay in the blood for very long. Okay, lower in vivo expansion. They don't divide in the cell and expand. Once you give CAR T, they just keep on dividing. They are okay. And there is also problem in making CAR NK cells. The way the whole thing. Look at this thing, days and weeks versus months and years for CAR T. Okay, so what we are working on, researchers are working on improving NK therapy. So just to get to, I keep on saying allogenic and autologous. So autologous is when the patient's T cells are taken out and given back to that patient. That is autologous, no rejection. Allogenic is that you take it from healthy, you make tons of CAR T and then you give to patient. That is allogenic. So that is hard for CAR T cell therapy. Okay. Now let's go to the macrophages. We have made CARs in macrophages too. So macrophages are those immune cells which will go to the site, eat up the whole garbage and clean it up. So we have made CAR T cells, CAR M cells. So here you do exactly the same thing. You take macrophages and then you express the CAR receptor there. Okay. So the beauty of macrophages is that they are very motile. They run to the tumor site, they eat it up. So when you make macrophages CAR, they enter solid tumors easily. Okay. After this, remind me, I will tell you about oncolytic viruses also. Because yesterday we were discussing why oncolytic viruses are not so commonly used. So just remind me after this. So these macrophages CARs, they enter solid tumors easily, they eat up the cell, they remodel the tumor microenvironment, and they activate the adaptive immunity. So they are really powerful macrophages, CARs. These are one of the recent discoveries. Okay, and once they activate it, all of these cells are recruited, and once they are recruited, there is a strong immunity. So see, macrophages eat pathogen, and these are expressing chimeric antigen receptor, and then they eat up, they release these cytokines, all of these guys come in, and once they came in, these T cells are activated, and they will kill the tumor cells. Okay, so these are CAR-M. Turn cold tumors into hot tumors. So again, cold tumors don't have, immune cell infiltration. So macrophages will go, and now they will recognize the tumor cell. Spelling is wrong. Okay, so cytokines are released, and then it will infiltrate the tumor cells. So this is mechanism of action. CAR-M cells are engineered. This is the CAR. They will target the tumor cells. So this receptor will target the tumor cells. So it binds there, and then it will eat up the tumor. It will eat up the cells in the tumor, and then it will secrete cytokines which will recruit all of these guys, and there will be enhanced tumor response, killing the tumor cells. Okay, so what are the advantages of CAR-M therapy? Better tumor infiltration, that is very important. Oncolytic virus cannot get into the tumor, that is the biggest problem. Multi-pronged anti-tumor mechanism, they are eating up the tumor, plus they are recruiting all the immune cells to kill the tumor. So it's a multi-pronged approach. This is CAR-M also have much more safety profile. You can imagine, these are going and eating up the tumor cells. They are not lysing it up and spreading all the garbage, they are just eating up the whole mess there. Okay. And this works for allogenic therapy also. Macrophages, you can put it in the body and there is not such a strong reaction. So they are supposed to have a broader therapeutic potential. So if you compare CAR-T with CAR-M, okay, so CAR-M, these things don't get into solid tumors. So there are many therapies which do not work very well for solid tumors because they cannot get inside that solid tumor pretty well. Oncolytic viruses are one of them. Oncolytic viruses only work for surface tumors like skin tumors and things like that. If you give oncolytic virus to patient, immune system clears up the virus completely. So there is no virus left to reach. Plus, even if it reaches a site, it cannot penetrate the tumors. That's why it has very limited efficacy despite being a very good idea. Okay. So dense stroma, dense extracellular matrix, macrophages, they eat up the ECM so they can get it in. Anything on the way, they eat it up. Okay. Immunosuppressive tumor microenvironment, you cannot stop macrophages from immunosuppressive tumor. They actually make the immunosuppressive thing to more immune responsive. So they will change it to hot. Low antigen density, no matter how low antigens are expressed, macrophages will eat it up. Okay, so CAR-M, and then they will recruit all of these immune cells. So they are very powerful. So CAR-T cells are just killing them. They are killing and eating them. Okay, then TME entry is poor. This is excellent. Antigen presentation weak. This is strong. This is best in blood cancer. CAR-T is exceptionally useful for blood cancer. These are very good for solid tumors, and they have more toxicity, they have less toxicity. So CAR-M is really becoming very powerful. Now let's go to the last one, which is universal CAR-T. So yesterday we were talking about CAR-T is so expensive, right? $500,000. It is personalized. So how we can make it universal? That we make it in a shelf, keep it in a pharmacy or in a hospital. Anyone who needs it, you give it to them. So if you have to make it universal, what is the thing you would like to do to a CAR-T? What is the problem? Why can't you give T cells from one patient to the other patient? Because of? because of rejection, and which is due to MSC, which is called HLA in humans. So if you knock down HLAs, it should work. And then there is this TCR, which is T cell receptor, because CAR T means T cells. And T cell receptors can also recognize, so you remove TCR and HLA and you're good to go. And then you can make universal. Do you guys agree with me? That's what it is. Okay. So see, you are removing TCR, endogenous TCR. TCR is T cell receptor. So you are basically taking T cells, right? And then you are expressing CAR. So CAR is different because that's your creation. But it also has endogenous TCR, T cell receptor, right? You remove those. It's a piece of cake to remove it. You just add the shRNA or CRISPR and you remove it. Okay, so we remove, and then we remove MSC1, which is HLA. Okay, and once you remove it, then these are universal CAR T. Why do we need universal CAR T? Because then you can take it from a healthy patient, make it and keep it and ready to go. Okay, so universal CAR T aims to create ready-made, standardized, rapid, accessible cell therapy. So how universal CAR therapy works, you remove endogenous TCR, you add the CAR, then you remove HLA molecules to prevent rejection, you add protection molecules, so you also add few other molecules. So if you add HLA, E and G, then it will, this is very interesting, explain to me what that means. Okay, so you remove some HLAs and then you add these HLA, E and G to avoid NK mediated rejection. What does it mean? Can you explain to me? So how do NK cells recognize self versus foreign? By HLAs or MSC. So those MSCs which are recognized by NK, you put them back. So NK say, oh, this is self, right? Although it is foreign, but you're basically fooling NK cells as self, right? And you add all of these things, and with CRISPR, it is a piece of cake. Talons and this ZFN are also editing methods, not as popular as CRISPR, but all of these things are relatively much easier. So then it is ready to go. I could not resist. I wanted to give you some background about MSC and HLA because you guys have some diverse background. So I have added there. So MSC and MSC2 are the general name for these molecules in all vertebrates. This is just for your knowledge. In humans, they are called HLA. And these are MSC class 1 have A, B, C HLA. MSC class 2 have DP, DQ, DR. Okay. All HLA molecules are human. All HLA molecules are actually human MSC molecules. HLA is just the human nomenclature for MSC molecules. So this is what it is telling you, and this is telling you that MSC stands for major histocompatibility complex. It is present in all nucleated cell, MSC class 1. Class 2 are present in APC cells. Name one APC cell to be. Dendritic cells. And then by the way, B cells are also APCs. Okay? Okay, so, and then HLA, you can see these are our HLAs. So key points, now you can understand the difference. When you call HLA, it becomes human, but they are actually MSC1. So this is all for your knowledge, that you don't get confused between MSC1, 2 and HLAs. So summary, HLA molecules are the main target of allo recognition. Our HLAs are different from each other, okay? So if you remove them, there is nothing there. So knocking them out reduces immune-mediated complications in allogenic therapy. This is the key strategy for universal CAR T therapy. Knock out HLA, hide donor cells from host immune system. Okay? All right. This is just everything I have told you, just very complicated way, but it is all listed here. We have discussed every single thing. Okay? So now you know what is the best way to do it. You make CAR T, you remove TCR, you remove the HLA which is causing rejection, and then you can just keep it in the freezer and infuse it as needed. So this can become a lot cheaper, okay? Now we come to, so the title of this was NK therapy and dendritic. So we have to finish with dendritic cell therapy in the next couple of minutes. So dendritic cells, you guys know, these are APC cells. So dendritic cell therapy is a type of immunotherapy that uses patients' own dendritic cells to train their immune system to fight diseases like cancer or autoimmune diseases. So you isolate dendritic cells, and then what do dendritic cells do? They recognize the antigen. So you take them out, and outside you train them to recognize the antigen, and once they are really primed, you put it back in the patient, and so that they can go and kill the tumor cells. So what they are doing is that dendritic cells recognize the antigen. So PAMP and DAMP, PAMPs are pathogen released. I've written what they are called. These are pathogen released antigen. DAMPs are damaged cell released antigens. So they are recognized by dendritic cells. They release cytokine, recruit all immune cells. Or they are APC cells, dendritic cells, so they will present in MSC, TCR will recognize it, and then T cell will be activated and B cells will be activated, and either T cells will kill it or they will activate B cells to release antibodies to clean up the foreign particle. So this is dendritic cell. So when it recognizes viral infection, it gets activated. Once it is activated, now this is very interesting. The dendritic cells will take it to the lymph node. Why it will take it to the lymph node? Because B and T cells live in lymph nodes. Okay, so suppose two really great people, like two very powerful group of people live there. So T cells live in T cell zone, B cells live in B cell zone, and they have a boundary. So what will happen is that these dendritic cells will go to the T cell area, and then they will present it to T cells, and T cell will get activated. So then these T cells who are activated, now they will migrate towards B cells, but they will not go inside B cell, they will wait in the boundary. At the same time, what happens, so B cells, they recognize the three-dimensional structure of the virus. They will not take the chewed up peptides. So they need the whole thing. So there are another kind of dendritic cells which are called follicular dendritic cells. They will present the whole thing to B cells by going to B zone in the lymph nodes. So B cells will recognize this. They will take it inside. They will chew it up. Now they are ready to make the antibody, but they need a second signal from the T cells. So now they will go towards T cells. T cells are going towards B cell. So where T and B cell boundaries there, they will meet. So B cells will present the chewed up stuff to them. T cells are already primed because they have received signal from dendritic cells. So T cells say, oh, okay, they will lock on and they say, you're good to go now. And then B cells will mature to plasma cells and release antibodies. Is it making sense? Should I repeat the whole thing? Maybe one more time, quickly. Okay, so what happens is that dendritic cells sense viral infection. Okay, then they take this, chew it up, and then dendritic cells will go to the lymph nodes. Lymph nodes are where B and T cells reside. So let's say half is B cell, which is B cell as a T cell zone, half of it. It's not half and half, but I'm making it easy, and this is B cell zone. B cells live in their zone, T cells live in their zone. So dendritic cells don't go to B cell, they go to T cells, and they will present, they are APCs, so they will present the chewed up stuff there. T cells will get activated. They will come to the meeting point for B cells. Here B cells independently got the virus, which was presented to them by another class of cells. So they now take the virus in, they chew it up, and they know that there is something wrong. But as a fail-safe mechanism, they have to get permission from T cells. So T cells are already primed, B cells chewed up, they have taken the virus, chewed up, they come and meet T cells. They both connect together, like actual connect together. T cells say, good to go. That's when B cells will become plasma cells, and then they will secrete antibodies towards that antigen. Make sense? That is the mechanism. Okay? So this is more complicated than what I told you. See, AI cannot make a perfect thing. I was struggling, I was feeding thing. Then I realized I can tell you better and you can figure this part out. Some part is correct, some part is a little bit here and there. Okay, if it was Purdue, I would make it myself with all the tools I had there. But here I was waiting for that. Okay, so this is just telling you that you can also make differentiated dendritic cells. So how do we do dendritic cell therapy? I got sidetracked. I was just telling you how antibodies are made, but let's come back to dendritic cell therapy. We take monocytes. Monocytes can be converted back to dendritic cells. So you give them these cytokines. They are mature. Now you expose them to tumor antigens. Now they are ready for those tumor. Now you put them in lymph nodes. They activate T cells. And then once the T cells are activated, they will kill the tumors. Okay, so that's the whole process here. All right, this is yet another picture of showing how they go and talk to this V zone and T zone. All right.